Hi everyone, I'm very glad to share our study, Convergent Evolution of Metabolic Functions, Evidence from the Gut Microbiomes of Humans and Dogs. Along with long-term cloning, dogs and humans have shown convergent evolution in their genomes, such as copy number variations in genes related to adapting to a starch-rich diet. Studies have shown that compared to mice and pigs, the composition of the gut microbiome in dogs and its response to dietary changes are more to those in humans. In our study, we collected fecal samples from 3 groups of dogs with different lifestyles, 2 wolf groups, and macaques for metagenomic sequencing, then compared with human metagenomic data from public. The analytical workflow included quality control of raw data, metagenome assembly, metagenomes quality control, and dereplication. The resulting metagenomes were used for 2 main analyses: comparative analysis between groups and human metagenomes, and construct gut microbial communities community analysis, functional annotation, and inter-host group comparative analysis. Results indicated that the Zhou wolves had the lowest microbial detection rate among all groups. We compared the microbiomes of the 6 groups with published human metagenomes. Macaques showed the highest overlap with the human metagenome at the species level, followed by dogs. While at the strain level, dogs exhibited the highest overlap with the human gut microbiome, significantly higher than that of macaques. We hypothesized that dogs harbor microbial strains closer to those in humans than macaques do. We also constructed gut microbial community profiles for each group. PICO-A results revealed distinct microbial community patterns for each group. 2 wolf groups differed significantly, and 3 dog groups, while sharing some microbes, also showed unique patterns. Indigenous dogs appeared closer to humans in community structure than macaques did. However, upset R analysis showed that macaques shared the most microbial species with humans, followed by indigenous dogs. The dendrograms of microbial communities also showed that human and macaque communities clustered closely together, which consistent with their host phylogenetic relationship. However, the dendrogram of functional enrichment grouped dogs and humans on the same branch. The pairwise comparative analysis of metabolic pathways among 4 host species showed that dogs possessed the strongest capabilities in most carbohydrate metabolism, particularly starch and sucrose metabolism. This likely evolved as an adaptation to the human diet and environment. We speculate that the dog microbiome may have evolved enhanced starch digestion capabilities to compensate for the host's physiological limitations to adapt to new diets and environments. Further functional comparison revealed that working dogs showed significant enrichment in pathways involved in lipid metabolism and xenobiotic degradation, whereas pet dogs had stronger functions related to amino acid, carbohydrate, and energy metabolism. Notably, working dogs enriched pathways associated with human diabetic cardiomyopathy and Alzheimer's disease, while pet dogs enriched pathways related to Helicobacter pylori infection. These results highlight the impact of environment and lifestyle. Accordingly, we proposed that domestic dogs could serve as a potential model for studying human metabolic disease and microbial responses to environmental changes. In this study, we first compared the gut microbiomes of macaques, 3 lifestyle dog groups, and 2 wolf groups with human gut microbiomes. Comparative analysis of metagenomes at the species and strain levels revealed that the dog gut microbiome is more similar to humans than that in macaques. Further community analysis showed that macaques are closer to humans in microbial community structure and composition, consistent with host phylogeny. However, functional analysis demonstrated that the dog gut microbiome's function is more similar to humans, providing evidence for convergent evolution in metabolisms between dogs and humans. Further analysis of functional enrichment in working dogs and pet dogs revealed that lifestyle-induced metabolic and diseases differences in dogs mirror similarities to human responses to working environments.